Good morning, everybody. Welcome back to daily lectures from New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby. We're on to the subject of transgenic animals and the subject of molecular biotechnology. This is chapter 21. I repeat, the subject has to do with transgenic animals. Let me just give you a synopsis of the topics covered under that. There will be transgenic mice methodology, retroviral vector method, the DNA microinjection method, the engineered embryonic stem cell method, genetic modification with CRE-IOXP, recombination system, RNA interface, transgenesis with high-capacity vectors, transgenic mice applications, transgenic disease models, Alzheimer disease, using transgenic mice as test systems, conditional regulation of transgene expression, condition control of cell death. And we have cloning livestock by nuclear transfer, transgenic livestock, production of pharmaceuticals, production of donor organisms, disease-resistant livestock, improving milk quality, improving animal production traits, transgenic poultry, transgenic fish, summary and review questions. Right now, on the subject matter proper. When traditional breeding methods are used, many generations of selective matings are required to improve livestock and other domesticated animals genetically for traits such as milk yield, wool characteristics, rate of weight gain, and egg-laying frequency. At each successive generation, animals with superior performance characteristics are used as breeding stock. Eventually, high production animals are developed as more or less pure breeding lines. This combination of mating and selection, although time-consuming and costly, has been exceptionally successful. Today, almost all aspects of the biological basis of livestock production can be attributed to this process. However, once an effective genetic line has been established, it becomes difficult to introduce new genetic traits by selective breeding methods. For example, a strain with a newly discovered valuable gene may also carry deleterious genes that, after crossing, would diminish the existing genetically determined production levels. Thus, a completely new program of multi-generation crosses with rigorous selection procedures has to be initiated to ensure that a new breeding line retains both its original attributes and the new trait. Excuse me. Until recently, selective breeding was the only way to enhance the genetic features of domesticated animals. However, the combination of the successful transfer of genes in the mammalian cells and the possibility of creating genetically identical animals by transplanting nuclei from somatic cells into nucleated eggs, nuclear transfer, nuclear cloning, led researchers to consider putting single functional genes or gene clusters into the chromosomal DNA of higher organisms. Conceptually, the strategy used to achieve this end is simple. 1. A cloned gene is injected into the nucleus of a fertilized egg. 2. The inoculated fertilized eggs are implanted into a receptive female because successful completion of mammalian embryonic development is not possible outside of a female. Some of the offspring derived from the implanted cells' eggs carry the clone gene in all of their cells. Animals with the clone gene integrated in their germline cells are bred to establish new genetic lines. This approach has many practical applications. If, for example, the product of the injected gene stimulates growth, animals that acquire this gene should grow faster or require less feed. An enhancement of feed efficiency by a few percent would have a profound impact on lowering the cost of production of either beef or pork. During the 1980s, with considerable effort, the idea of genetically manipulating animals by introducing genes into fertilized eggs was converted into reality. As with many new scientific enterprises, a set of terms was created to make communication easier. For example, an animal whose genetic composition has already been altered has been altered by the addition of foreign exogenous DNA is said to be transgenic. The DNA that is introduced is called a transgene, and the overall process is called transgenic technology or transgenesis. The genetic improvement of animals by the introduction of relevant transgenes is only slowly being realized. In the meantime, however, transgenesis has become a powerful technique for studying fundamental problems of mammalian gene expression and development, for establishing animal model systems, for studying human diseases, for producing foreign proteins in birds' eggs, and for using the mammary gland to produce pharmaceutically important proteins in milk. With this last exception in mind, the term farming was coined to convey the idea that milk from transgenic farm animals can be a source of authentic human protein, drugs, or pharmaceuticals. There are a number of reasons why the mammary glands should be used in this way. Milk is a renewable, secreted body fluid that is produced in substantial quantities and can be collected frequently without harm to the animal. A novel drug protein that is confined to the mammary gland and secreted into milk should have no side effects on the normal physiologic process of the transgenic animal and should undergo post-translational modifications that at least closely match those in humans. Finally, purification of a protein from milk, which contains only a small number of different proteins, should be relatively straightforward. We're on to the topic of transgenic mice methodology. Transgenic technology has been developed and perfected in the laboratory mouse. Since the early 1980s, hundreds of different genes have been introduced into various mouse strains. These studies have contributed to an understanding of the gene regulation, tumor development, immunological specificity, molecular genetics of development, and many other biological processes of fundamental interest. Transgenic mice have also played a role in examining the feasibility, the industrial production of human therapeutic drugs by domesticated animals and in the creation of transgenic strains that act as biomedical records models for various human genetic diseases. For transgenesis, DNA can be introduced into mice by one retroviral vectors that infect the cells of an early stage embryo prior to implantation into a receptive female. 2. Microinjection into the enlarged sperm nucleus, male pronucleus of a fertilized egg. Or 3. Introduction of genetically engineered embryonic stem cells into an early stage developing embryo before implantation into a receptive female. The retroviral vector method of the various gene transfers. Methods. The use of retroviral vectors has the advantage of being an effective means of integrating the transgene into the genome of a recipient cell. Retroviruses have RNA genomes that are used as templates for reverse transcriptase to synthesize a DNA copy that can be inserted into the host cell genome. However, vectors derived from these viruses can transfer only small pieces, 8 kilobases of DNA, that because of the size constraint may lack essential adjacent sequences for regulating the expression of the transgene. There is a further major drawback to the use of retroviral vectors. Although these vectors are designed to be replication defective, the genome of the retroviral strain helps virus that are needed to create large quantities of the vector DNA can be integrated into the same nucleus as the transgene, despite special precautions. Helper strain retrovirus can be produced by the transgenic organism. Consequently, for applications in which either a commercial product is to be synthesized by the transgenic organism or the transgenic organism is to be used as food, it is absolutely necessary excuse me, that there be no retroviral contamination. In addition, transgenes introduced on some retroviral vectors are silenced in mouse embryos. Transgenes carried on vectors derived from lentiviruses, a group of retroviruses that includes human immunodeficiency virus and similar viruses from other animal cells, however, are not silenced in embryos. Moreover, these vectors are capable of delivering large sentiments of DNA into the host genome, are stable for relatively long periods, have low immunogenicity, and can infect both dividing and non-dividing cells. The last characteristic is an advantage for the expression of transgenes in neuronal, muscle, liver, and other non-dividing cells. The lentivirus vector system is similar to other retroviral vectors and is comprised of a transfer vector that carries the transgene and a packaging cell line that provides viral proteins for packaging the viral particles. Lentiviral vectors have been used successfully to introduce a variety of transgenes into embryonic cells or early embryos of mice, pigs, cattle, and birds. And these genes are highly expressed either ubiquitously or in a tissue-specific manner. Depending on the promoter used, lentiviruses are also often used as delivery vehicles to introduce a construct to decrease gene expression in mice and other organisms by RNA interference, RNAi, shown below. Now, the DNA microinjection method. Because of the disadvantages of the retroviral vector method, microinjection of DNA is currently the preferred method for producing transgenic mice. This procedure is performed in the following way. The number of available fertilized eggs that is to be inoculated by microinjection is increased by stimulating donor females to superovulate. Female mice are given an initial injection of pregnant male serum and another injection about 48 hours later of human chorionic gonadotrophin. A superovulated mouse produces about 35 eggs instead of the normal 5 at a time. The superovulated females are mated so that eggs become fertilized, and then they are killed. The fertilized eggs are flushed from their oviducts. Microinjection of the fertilized eggs usually occurs immediately after the collection. The microinjected transgene construct is often in a linear form and free of prokaryotic vector DNA sequences. In mammals, after entry of the sperm into the egg, both the sperm nucleus, male pronucleus and female nucleus are separate entities. After the female nucleus completes its meiotic division to become a female pronucleus, nuclear fusion, karyogamy, occurs. The male pronucleus, which tends to be larger than the female pronucleus, can be located by using a dissecting microscope. The egg can be maneuvered, oriented, and held in place by micromanipulation while the DNA is microinjected. On a good day, several hundred male pronuclei can be nucleated. The procedure, although apparently simple, requires the coordination of a number of experimental steps. Even a highly trained practitioner can expect, at best, only 5% of the nucleated eggs to develop into transgenic animals. None of the steps in the procedure is 100% efficient. Consequently, large numbers of micro-injected fertilized eggs must be used. Furthermore, with this method, the injected DNA integrates at random sites within the genome, and often multiple copies of the injected DNA are incorporated at one site. Not all the transgenic pups will have the appropriate characteristic. In some individuals, a transgene may not be expressed because of the site of integration, and in others, the copy number may be excessive and may lead to overexpression, which disrupts the normal physiology of the animal. Despite the overall inefficiency, it has become routine to use DNA microinjection of the male pronucleus to create lines of mice carrying functional transgenes. The engineered embryonic stem cell method. Cells from the early blastocyte stage of a developing mouse embryo can proliferate in cell culture and still retain the capability to differentiate into all of the cell types, including germ-line cells, after they are reintroduced into another blastocyte embryo. Such cells are called pluripotent embryonic stem cell. While in culture, embryonic stem cells can be readily engineered genetically without altering the pluripotency with the system. For example, a functional transgene can be integrated at a specific site within a dispensable region of the genome of an embryonic stem cell. The genetically engineered cells can be selected, grown, and used to generate transgenic animals. In this way, the randomness of integration that is inherent in the DNA microinjection and retroviral vectors is avoided. After transfection of embryonic stem cells in culture, with a DNA vector that is designed to integrate within a specific chromosomal location, some cells will have DNA integrated at non-target spurious sites, whereas in other cells, integration will occur at the targeted correct site. The target site should be located in a section of genomic DNA that encodes no essential products, so that after integration of the input DNA, there is no interference with any developmental or cellular functions. Moreover, it is essential that this transgene be integrated into a part of the genome that does not prevent it from being transcribed. For example, in euchromatin rather than heterochromatin. In most of the embryonic stem cells, the input DNA will not be integrated at all to enrich for the cells with DNA. Integrated at the target site, a procedure called positive and negative selection is implemented. This strategy uses positive selection for cells that have vector DNA integrated anywhere in their genomes. And a negative selection against the vector DNA sequence is that is integrated at spurious sites. A targeting DNA vector for the positive and negative selection procedure usually contains one or two blocks of DNA sequences, HP1, HP2, that are homologous to separate regions of the target site. 2. The transgene, which will confer a new function of this recipient. 3. A DNA sequence that codes for resistance to the compound G418-NEO. And 4. Two different genes for thymidine kinase TK1 and TK2 from herpes simplex viruses types 1 and 2. HSV TK1, HSV TK2. The arrangement of these sequences is key to the positive negative selection procedure between the two blocks of DNA that are homologous to the target site of the genes for the transgene G418 resistance NEO gene. Inside of each of these homologous blocks are the genes HSV TK1 and TK2. If integration occurs at a spurious site that is not at BHV1, HP2, either one or both of the HSV TK genes have a high probability of being integrated along with other sequences. Alternatively, the integration event is due to homologous recombination by a double crossover at the target site. The HSV TK genes are excluded and only the transgene and the NEO gene are incorporated into the genome. When transfected G cells are grown in the presence of G418, all the cells that lack the NEO genes are killed. Therefore, these cells with integrated DNA survive, that is, these cells are positively selected. If the compound Gansi-Clover is added to the same time as G418, the cells that express thymidine kinase are killed because thymidine kinase converts Gansi-Clover into toxic compounds that kill cells. That is, these cells are negatively selected. The cells most likely to survive the stool selection scheme are those that have DNA integrated at the target site. Although not foolproof, the positive-negative selective method enriches an embryonic stem cell population for cells to carry a transgene at a specific chromosomal location. A more direct way to detect embryonic stem cells that carry a transgene at a targeted chromosomal site is to use PCR. The targeting DNA vector contains two blocks of DNA that are homologous to the target site, with one on either side of both the transgene and the cloned bacteria or synthetic unique DNA sequence that is not present in the mouse genome. After the transfection of embryonic stem cells, the cells are pooled and samples are screened by PCR. One of the primers P1 for PCR is complementary to a sequence within the cloned bacterial or synthetic unique DNA sequence of the integrating vector. The other primer P2 is complementary to a DNA sequence that is part of the chromosome adjacent to the region of one of the homologous blocks of DNA. If integration is at a random site, the predicted amplified DNA product is not synthesized. However, if site-specific integration occurs, the PCR amplifies a DNA fragment of known size. In this way, pools of cells with embryonic stem cell contains a desired gene. The targeted site can be identified by subculturing from these pools. Cell lines carrying the site-specific integration can be established. Embryonic stem cells carrying an integral transgene can be cultured and inserted into blastocyte stage embryos. These embryos can then be implanted into pseudo-pregnant foster mothers. Transgenic lines are established by mating the progeny that carry the transgene in the germ lines. Then, if required, littermates that carry a transgene in the germ lines are crossed to produce mice that are homozygous for the transgene. Not only can a transgene be inserted into a specific chromosome site by homologous recombination and embryonic stem cells to provide a new function, but a specific mouse gene can also be targeted for disruption by the incorporation of a DNA sequence usually a selectable marker gene, into its coding region. One of the aims of targeted gene disruption, gene knockout, is to determine the developmental and physiological consequences of inactivating a particular gene. In addition, a transgenic lean with a specific disabled gene can be used as a model system to study the molecular pathology of a human disease. For example, inactivation of the mouse rhodopsin gene by targeted gene disruption leads to deterioration of the rod cells and mouse rhodopsin gene by targeted gene disruption leads to deterioration of the rod cells of the retina in transgenic mice that closely resembles a human disease, retinitis pigmentosa. Thus, the progress of retinal degeneration and the effects of potential therapeutic agents that either delay or block the genetically induced retinopathy have been studied by using the rhodopsin knockout mouse. Hundreds of different types of knockout mice have been created as animal models for the study of various human abnormalities. Genetic modification with the KORAH-BIOTECH recombination system. The bacteriophage B1 of several tailed viruses that infect E. coli. It has a double strand of DNA. Genome that is about 100 kb in length. After introduction into E. coli, the linear B1 genome forms a circle. The circularized B1 DNA acts as a template for replication, and depending on which set of genes is activated, the circular form is either maintained as a plasmid or used as a template for the production of viral genomes during the life cycle. On rare occasions, the circularized B1 genome integrates to the E. coli chromosome. Circularization and integration of the P1 genome are mediated by the product of the CRE gene, circularization recombination CRE recombinase protein, which specifically cleaves and recombines the DNA of LOXP, locus of crossing over X in P1 sites. A LOXP site consists of two 13 base pair inverted repeats that are separated from each other by an 8 base pair spacer sequence. Briefly, KORAH-LOXP recombination entails the coming together of two remote LOXP sites, each of which has two bound KORAH recombinase molecules, cleavage by the KORAH recombinase within the spacer regions between the repeat sequences, and the exchange and joining of DNA strands to form recombined DNA molecules. The outcome of the recombination event depends on the orientation of the repeats of the LOXP sites. If the repeats are in opposite directions, then the exchange inverts the DNA between the two LOXP sites. If the repeats are in the same orientation, then the intervening sequence is excised. The repeat elements of bacteriophage B1 are naturally in opposite orientations. The KORAH-LOXP recombination system can function when the LOXP sites are widely separated. The outcome of the recombination event depends on the orientation of the repeats of the LOXP sites. If the repeats are in opposite directions, then the exchange inverts the DNA between the two LOXP sites. If the repeats are in the same orientation, then the intervening sequence is excised. The repeat elements bacteriophage B1 are naturally in opposite orientations. The KORAH-LOXP recombination system can function when the LOXP sites are widely separated. For example, the two LOXP sites that are essential for circularization of a P1 genome are about 100 kb apart. The specificity of the KORAH-LOXP system is absolute because the recombinase acts exclusively on LOXP sites. On the basis of these features, the KORAH-LOXP system was developed for producing cell-specific gene modifications in mouse cells. As a first step in the overall strategy, the KORAH gene is isolated and replaced under the control of a cell-specific promoter. Transgenic mice with the KORAH gene construct are established, and the tissue specificity of the KORAH activity is confirmed. Next, the LOXP site with the repeat sequences in the same side direction is inserted on either side of a cloned DNA sequence, such as a cloned exon. The construct is integrated into a chromosome site of embryonic stem cells by homologous recombination. These cells are selected, cultured, and used to establish transgenic mouse line. Then a transgenic mouse with the tissue-specific CSIR transgene is mated with the transgenic mouse with the integrated LOXP flank sequence. The DNA between the two LOXP sites is deleted after the KORAH transgene is expressed in double transgenic organisms. And this way, the biological consequences of the loss of activity of a gene in a specific tissue can be monitored. The KORAH-LOXP recombination system can also be used to activate a transgene in a specific tissue. In this case, the sequence between LOXP sites prevents transcription. This construct is inserted between the promoter and the coding sequence of a transgene. When KORAH is expressed in mice with the integrated construct, the DNA sequence that blocks transcription is excised, thereby enabling the expression of the transgene. By controlling expression of KORAH, for example, by adding an inducer that activates the KORAH promoter to the drinking water of mice, the expression of a transgene can be controlled. The KORAH-LOXP technology has been used extensively to study the biological consequences of tissue-specific gene inactivation, with the goal of establishing models for human diseases. For example, selective removal of the kinesin 2 gene, which is expressed exclusively in retinal photoreceptor cells, leads to an accumulation of opsin and arrestin, and eventually to cell death. This result mimics aspects of inherited retinitis pigmentosa in humans and is used for detailed studies of the pathophysiological effects on the retina. Larger chromosomal aberrations, such as deletions, can also be created with the KORAH-LOXP system in humans. A large deletion within chromosome 22 is associated with the DiGeorge syndrome (DGS), which is cardiovascular dysfunction, as a significant characteristic. It is not known whether DGS is due to the loss of a large number of genes for a few major ones. Now, to determine the basis of DGS, a larger deletion of the mouse chromosome that is comparable synthetic to human chromosome 222 was generated with the KORAH-LOXP excision strategy. The mice with this deletion had symptoms that resembled DGS. Moreover, when a transgene for a cardiovascular specific transcription factor for this region was introduced into these mice, the DGS-like effects were partially overcome, which suggests that the loss of this gene in humans plays a key role in DGS. RNA interface. The two main methods to silence gene expression in animal cells to study biological processes. One method abolishes expression of a gene by targeted disruption through homologous recombination in embryonic stem cells knockout method. And the other decreases the expression of a target gene knockout method knockdown method by preventing messenger RNA, mRNA translation using RNAi. The latter method exploits a natural mechanism for regulation of gene expression by endogenous RNA molecules in animals and plants, and for protecting cells against exogenous RNA molecules from invading viruses. And RNAi, double-stranded RNA, is recognized by ribonuclease RNase called dicer that cleaves the RNA into smaller double-stranded RNA molecules known as small interfering RNA, siRNA. A large nucleus complex called RISC RNA induced silencing complex. We've heard these terms before, right? So try to recollect, and we get to know how well you read your portions, right? Separates the strands of siRNA as silent RNA and the silencing complex. And the single-stranded RNA products, together with RISC, bind to homologous sequence on mRNA molecules. The nuclease compound on RISC then degrades the mRNA, which prevents the encoded protein from being synthesized. Short endogenous RNAs, microRNAs transcribed from regions of the genomes of animals and plants, also recognized by dicer and RISC and block translation of a target mRNA. To create transgenic mice with a reduced expression of a target gene, a small region of the target sequence is cloned into a vector as an inverted repeat, separated by a short spatial region. The RNA transcript that is produced from the sequence forms a short 19 to 21 base pair hairpin RNA, small or short hairpin RNA, shRNA, due to intramolecular base pairing. Excuse me. Shorter sequences are used to avoid a general down-regulation and translation that is often elicited with longer sequences, though not to occur as a part of the viral defense system. The construct is introduced into mouse embryonic stem cells by pronuclear injection or on a lentiviral vector. Stable mouse lines have been generated that produce an shRNA that is processed by the cells, animals' dicer and RISC proteins into siRNA to reduce the expression of a target gene. This expression has been applied to a variety of anti-males, including cows, pigs, goats, frogs, and rats. In cases in which reduced expression of a target gene might impair the growth and development of the animal, the timing of expression of the shRNA can be controlled. The conditional knockdown blocking sequence is inserted into the vector between the sequence encoding the shRNA and the promoter that controls expression of the hairpin RNA. The blocking sequence carries a terminal signal for RNA polymerase and can also contain a marked gene such as GFP. Encoding gene fluorescent protein under the control of its own promoter, the blocking sequence is flanked by two lock-speed sites in the same orientation and therefore is excised following the induction of recombinase. Excision of the blocking sequence restores expressions of the hairpin RNA and downregulation of the target gene in the mouse genome. Excuse me. Transgenesis, where the high-capacity vectors to generate transgenes are complementary DNAs, cDNAs. Small genes, less than 20 kilobase pairs, or parts of genes, often cDNAs, are poorly expressed in mammalian cells. Also, when a segment of genomic DNA is used for transgenesis, important gene-specific regulatory sequences that lie either upstream or downstream of the gene are rarely retained as part of the insert. Moreover, complex genes and multi-gene complexes are too large. We now have a milestone note. Genetic transformation of mouse embryos by microinjection of purified DNA, courtesy J.W. Gordon, G.S. Ken Ghosh, D.J. Plotain, G.A. Barbosa, and F.H. Ruddle. Thank you to all of them. This is a publication that appeared in 1980 in National Academy of Science. And the other topic as well is somatic expression of herpes thymidine kinase in mice following injection of fusion gene into eggs. Here again, courtesy to Arl Brinster, H.Y. Chan, M. Trumblaw, W. Senior, Warren, and Pomita. Now we're going to the topic proper. Gordon Toll with the first to show the feasibility of DNA transfer by microinjection into the pronucleus of the mouse egg. In this study, the procedure was tested by microinjecting several hundred eggs with a vector gene construct that consisted of PBR222, carrying both the HSV thymidine kinase and a piece of the simian virus 40 genome. Of the 78 offspring that were obtained from the surrogate mothers still retained some plasmid DNA, the authors concluded. These data demonstrate that it is possible to use a recombinant plasmid as a vector to transfer foreign genes directly into mouse embryos, and that these embryos can maintain the foreign genes through the development. Unfortunately, the plasmid DNA was not intact, and the HSV thymidine kinase sequence did not become a transgene. On the other hand, Brinster et al., who microinjected the pronuclei of a number of mouse eggs with a plasmid carrying the HSV thymidine kinase gene under the control of the promoter of the metallothionein 1 gene, found that one of the transgenic mice expressed HSV thymidine kinase at a high level in its liver and kidneys in comparison to 3 other transgenic mice that produced low levels of this enzyme. Also, 8 other transgenic animals carried the HSV thymidine kinase sequence but did not produce any active HSV thymidine kinase. Southern blot analysis showed that all the transgenic mice contained multiple copies of the microinjected DNA. These studies laid the foundation for transgenesis of mice. Despite the technical complexity and relative inefficiency of the microinjection strategy, it's been exceptionally successful. Currently, some scores of strains of mice with either foreign genes, transgenic mice, or endogenous genes that have been disrupted by the insertion of foreign DNA in knockout mice are being used for studying gene regulation, mammalian development, viral pathogenesis, cancer toxicology, and the mutagenicities of various agents, among others. In addition, transgenic and knockout mice are of considerable biological importance as model systems for human diseases. For conventional vectors here, for these reasons, high-capacity vectors that carry genomic DNAs range in size. From 100 to more than 1,000 kilobase pairs have been developed for transgenes. These vectors have been derived from bacteria, P1 bacteriophage derived mammalian and yeast YAC's artificial chromosomes. A number of transgenic mice have been produced by microinjection of the pronucleus of the fertilized egg or transfection of embryonic stem cells with YACs that carry either an array of related genes or a single large gene. These organisms have been used to study developmental processes as models for human disorders with the production of human therapeutic agents. The production of mice, that synthesize only human antibodies is another noteworthy example of YAC transgenesis. In theory, monoclonal antibodies can be effective agents for diminishing the proliferation of cancer cells and as a means of treating other diseases. However, it is possible to generate human monoclonal antibodies routinely. Also, unfortunately, rodent monoclonal antibodies are immunogenic to humans and elicit anti-mouse antibodies and result in a destruction of therapeutic antibodies and sometimes cause allergic reactions. Recombinant DNA strategies have been devised to humanize existing rodent monoclonal antibodies. An antibody is a tetrameric protein with two pairs of dissimilar chains. One of the chains is called the heavy chain, and the other is called light chain. The terms heavy and light refer to the difference in the molecular masses of the antibody subunits. The genetic information for a specific heavy chain is created by rearrangement of several heavy chain specific DNA segments in a B cell, an antibody producing cell. In addition, there are two different types of antibody light chains that are encoded after DNA rearrangements of other light chain specific DNA segments. Each single B cell synthesizes only one kind of antibody molecule that has a unique set of rearranged segments for a heavy chain and a light chain. The genetic repertoire for the formation of the vast numbers of different human antibodies consists of more than 100 heavy chain DNA segments and a similar number of light chain DNA segments. Each heavy and light chain gene locus is about 1 to 1.5 megabase in length. To create a transgenic mouse that is capable of synthesizing a full range of human antibodies against every antigen, the endogenous mouse heavy and light chains genes were inactivated. YAC is carrying most of the heavy and light chain DNA elements from each human immunoglobulin gene were inserted into the chromosomal DNA of the mouse. A commercialized version of the human antibody producing mouse has been designated the Xena mouse, and the first fully human monoclonal antibody produced in this mouse, panitumumab, has received regulatory approval for use as a treatment for advanced colorectal cancer. Clinical trials have shown that panitumumab, an effective control agent for colorectal cancer, and does not elicit production of anti-panitumumab antibodies. The development of this cancer treatment took approximately 15 years to regulatory approval, including the development of the Xena mouse. Other therapeutic antibodies produced in the Xena mouse, including several for the treatment of various cancers and osteoporosis, are now in the clinical trials. Transgenic mice applications. Transgenic mice can be used as model systems for determining the biological basis of human diseases and devising treatments for various conditions. In addition, transgenesis of mice is an exemplary system for proving whether the production of a potential therapeutic agent is feasible. The whole animal models simulate both the onset and progression of a human disease. However, a mouse is not a human even though it is a mammal, and so the information gathered from some transgenic models may not always be medically relevant in other instances. However, critical insights into the etiology of a complex disease can be gained. With this in mind, mouse models for human genetic diseases such as Alzheimer disease, amyotrophic lateral sclerosis, Huntington disease, arthritis, muscular dystrophy, tumorigenesis, hypertension, neurodegenerative disorders, endocrinological dysfunction, and coronary disease, as well as many others, have been developed. Transgenic disease models and Alzheimer's disease. Alzheimer's disease is a degenerative brain disorder that is characterized by the progressive loss of abstract thinking and memory and is accompanied by personality change, language disturbances, and a slowing of physical capabilities. Clinical diagnosis of Alzheimer's disease is poor, although 1% of the population between 60 and 65 years of age and 30% of the population over 80 years of age may develop it. Neurofibrillary tangles accumulate within the cell bodies of the neurons. Dense extracellular aggregates called senile plaques develop at the ends of inflamed nerves neuritis, and brain cells, neurons, are lost in the neocortex and hippocampus of the brain in patients with Alzheimer's disease. The core of a senile plaque is composed of closely packed fibrillar structures that traditionally has been called an amyloid body. Originally, amyloid bodies were thought to be made up of carbohydrates, but more definitive analysis established that they are protein aggregates. However, despite the misnomer, the term amyloid has been retained. The principal protein of Alzheimer's disease amyloid bodies is a 4-kilodalton protein called A-beta amyloid beta or beta protein, beta amyloid protein or beta A4. The A-beta protein ranges in length from 39 to 42 amino acid residues. The A-beta 40 and A-beta 42 forms are the main variant. All A-beta proteins are derived from an internal proteolytic cleavage of the beta amyloid precursor protein, APP. Faulty cleavage of the APP protein causes the production of A-beta 40 and A-beta 42 and inefficient clearance of the variants likely lead to their accumulation. A small number of families with a high incidence of Alzheimer's disease have mutations in the APP gene. A finding that implicates this gene in the disorder. Unfortunately, for the most part, it is possible to study the onset and pathogenesis of Alzheimer's disease. It is an invaluable research tool. The mouse models for Alzheimer's disease were created with transgenes that contain mutations in the APP gene that occur in some families with a high incidence of early onset before 50 years of age of Alzheimer's disease. In one set of these families, site 717 APP 717 contains phenylalanine instead of valine. In another group of families with Alzheimer's disease sites, 670 to 71 of APP contain asparagine and leucocene instead of lysine methionine. A transgene with the APP 717 mutation was constructed from an APP cDNA modified introns were added between exons and 6 and 7 and 7 and 8 and 8 and 9 of the APP cDNA. The introns were introduced in the APP DNA because experiments show that transgenes with introns have an increased rate of transcription in comparison to constructs without them. The APP cDNA intron construct is controlled by the platelet-driven growth factor beta promoter that is expressed in brain tissue. The complete construct is called the PD-APP minigene, aging transgenic mice more than 6 months old, with about 40 copies of the PD-APP minigene display amyloid plaques, neural cell death, and memory defects. An APP 670/671 gene construct that is driven by a brain-specific promoter also produces transgene mice with Alzheimer's disease-like features. This lecture should be over in a minute and a half from now, and I'll continue in the next lecture. Till then, please make notes and take adequate screenshots of necessary figures as you see through. Right? Excuse me. Aging transgenic mice, more than 6 months old, with about 40 copies of the PD-APP minigene display amyloid plaques, neuronal cell death, and memory defects. An APP gene construct that is driven by a brain-specific promoter also produces transgenic mice with Alzheimer's disease-like features, including an excess of A-beta 42. Interestingly, neither aging PD-APP minigene mice nor APP 670/71 transgenic mice have neurofibrillary tangles. Possibly these structures are a secondary response to the overproduction of A-beta 42 in humans. The formation of amyloid Plaques in humans has also been shown to be associated with increased production of protein BACE1 beta site APP cleaving enzyme 1, the protease that cleaves APP to produce A beta, transgenic mice that produce, so this lecture should be winding up now,